Okay, so welcome back to this next video in which we are discussing phosphoinositide 3-kinases of the type 1. Okay, right. So, in this video what we're now going to do is look at the structure of type 1a PI3 kinase enzymes. And remember, these consist of one of those three catalytic subunits. Okay, so P110 alpha, P110 beta, or P110 delta, partnered with one of these five regulatory subunits here. Okay, P50 alpha, P55 alpha, P85 alpha, P85 beta, and P55 gamma, one of those five. Okay, right, so we're now going to discuss the structure of each of these two subunits, the catalytic subunit and then the P85 type regulatory subunit, how the two interact with one another, and then how the receptor tyrosine kinase is going to activate the type 1a PI3 kinase enzyme. Okay, right, so we're going to start off with the type 1a PI3 kinase catalytic subunit. Okay, so we'll draw this here. So, remember, it's a single protein now. We're referring either to P110 alpha, P110 beta, or P110 delta. Okay, so this is going to be P110 alpha, P110 beta, or not P110 gamma, whoops, P110 delta there. Okay, so that should be a delta, not a gamma. Okay, right, so we're about to draw this subunit here. Right, so this is going to be the amino terminus of this protein here. Okay, now the first major domain that you have in the polypeptide, okay, so this line represents a polymer of amino acids, and now this box is going to represent a whole domain. Okay, so it doesn't just represent a linear polypeptide here, it represents the polypeptide folded into something fancy, and then it will come out of this to go on to the next domain. Okay, and this important domain here is known as the ABD domain. Okay, now what does that stand for? Well, the ABD domain stands for the adapter binding domain. Okay, right, now why is it called that? Well, it's the portion, well, it's one of the portions, as we'll see, that is going to interact with the regulatory subunit of the PI3 kinase enzyme. Now, the regulatory subunit is going to interact with the receptor tyrosine kinase, okay? So, you're going to have receptor tyrosine kinase becoming activated, then it's going to bind to the regulatory subunit, and then that binding is going to activate the catalytic subunit. So, effectively, the regulatory subunit is acting as an adapter. It's the protein in between the receptor tyrosine kinase and the catalytic subunit of the PI3 kinase enzyme. Okay, so that's why this region, which is binding to this adapter protein, is called the adapter binding domain. Okay, so let's colour in the adapter binding domain, or the ABD here, in blue. Okay, right. After that, the next important domain you're going to have, okay, so once again this is the polypeptide linker that's going to link these two domains here, okay, the next important domain you have is a portion known as the RAS binding domain, or the RBD, okay, so I'm conscious that I don't want to invade my space here too much because I'm going to need that later, so I'm going to put this name up here, okay, so the RBD stands for the RAS. RAS is a monomeric G protein. Now, these catalytic subunits can also directly bind to RAS monomeric G proteins, and this serves some role in their activation. Okay, we're not going to go into this in much detail because it's not understood to a great amount of detail, but it is recognized that this is an important mechanism by which overactivation of RAS can contribute to tumorigenesis, okay, because the usual pathway that people associate with RAS gain-of-function mutations is that it overactivates the RAS-RAF-MEK-ERK pathway. However, there is this other function of RAS that it also interacts with the PI3 kinase type 1a enzymes, and this will also activate a pathway that's pro-oncogenesis, basically. So in some cancers, it's actually more important that RAS is activating PI3 kinase enzymes than it is that RAS is activating RAF kinase enzymes. Okay, right, but we're not going to discuss RAS activation of the type 1a PI3 kinase enzymes, but I will note that that domain is here. Okay, so that's the RAS binding domain. Then, after the RAS binding domain, you have a domain known as the C2 domain. Now, this is also going to be involved in binding to the regulatory subunit. Now, this is a little bit odd because C2 domains are usually very important in calcium signaling. They're usually domains which bind calcium, but in this case, the C2 domain is going to bind to the regulatory subunit of the PI3 kinase enzyme. Okay, so this is a C2 domain in orange here. So I'll highlight that up in orange. Okay, so this is also going to be important in binding to the regulatory subunit, as we'll see in a moment. Okay, and it's just a conserved domain that you find in a huge number of different proteins, so many different proteins. A notable example is protein kinase C enzymes. Those have a C2 domain, so it's just a conserved structure that you can find within many proteins. Okay, now after the C2 domain, what you then have is a region known as the helical portion. Okay, and this is actually also going to be involved in binding to the regulatory subunit. Okay, so there are many different domains of this catalytic subunit of our PI3 kinase enzyme that are involved in binding to the regulatory subunit. Okay, and remember, this picture represents any one of these that you wish to choose. It's true for P110 alpha, P110 beta, and P110 delta. Then, after the helical portion, you then have the actual catalytic domain. Okay, so this is the portion that actually catalyzes the addition of the phosphate group onto the phosphatidylainositol 4,5-bisphosphate molecules, and then it ends with the carboxylic acid terminus there. Okay, so we'll colour in the catalytic subunit here in turquoise. Okay, right, so this is the structure then of a catalytic subunit of PI3 kinase type 1a enzymes. Okay, right, now what I want to do is show you the structure of one of these P85 type regulatory subunits. Now, this does vary between these different P85 type regulatory subunits. Okay, so you'll notice that we have some big P85 type regulatory subunits such as P85 alpha and P85 beta. Okay, so I'll highlight these up in a specific colour. So this one here and this one here, those are the big ones. And then we have these smaller ones here, P55 gamma, P50 alpha there, and P55 alpha there. Those are the smaller ones. Now, the smaller ones are going to lack two domains that the P85 ones have, as we'll see in a moment. Okay, but these two domains are not essential for the function of the regulatory subunit, and actually we don't have a clue what they're doing in these larger P85 alpha and P85 beta regulatory subunits. Okay, so we'll start with these two domains then that are present in those bigger regulatory subunits. Okay, because they're actually at the amino termini. Okay, so here's the amino terminus of our regulatory subunit protein. Okay, now the first domain that you have is a very famous domain that's present in a huge number of different proteins. It's known as the SH3 domain. Okay, so I'll colour in this SH3 domain in pink here. Okay, so what does this stand for? Well, SH3 stands for Sark homology free domain. Okay, so S is for Sark. Sark is a tyrosine kinase enzyme that's present within the cytoplasm. Okay, and we'll come back to tyrosine kinase enzymes in a moment. This is a free tyrosine kinase. It's not a receptor tyrosine kinase. Okay, and then free domain. So this domain, again, it's a structure that you find in a huge number of different proteins, not just the PI3 kinase type 1A regulatory subunits. Okay, and it was first identified in the tyrosine kinase enzyme SARC, and hence how it became known as the SARC homology free domain. There are loads of domains that were first discovered in SARC, and they're all the SH domains. Okay, right. So, SH3 is our first domain that is present in these big ones, but not in the small ones. And it's not essential. It doesn't seem to be essential. We don't actually know what it's doing in these regulatory subunits of PI3 kinase proteins. Okay, right. The next domain that you have after the SH3 domain is what's known as a breakpoint cluster homology domain. Okay, so this BH domain stands for breakpoint cluster homology domain. Okay, so the B is for breakpoint cluster, and then the H is for homology. Okay, right. So again, this is a domain that you find in many different proteins. It's just a conserved structure. Okay, so we'll colour this one in yellow here. Right, okay, so those two domains that we've now talked about, the SH3 domain and then the breakpoint cluster homology domain, these are the two domains that are not going to be present in the shorter ones. Okay, so these are present in these big ones, P85 alpha and P85 beta. Okay, so these are present in P85 alpha and P85 beta, but they are not present in the shorter ones, which remember are P50 alpha, P55 alpha, and P55 gamma here. Okay, so for the shorter ones, the amino terminus would start here, and the first domain then that you would have in those shorter ones would then be a domain known as the NSH2 domain. Okay, but you're going to have this domain after the breakpoint cluster homology domain if we're talking about one of these bigger P85 alpha or P85 beta proteins. Okay, right, so let's colour in the SH2 domain in, I think we'll have red. Okay, so this again is another one of these SARC homology domains, and this is going to be really important. It has a strong interaction with this helical domain of the catalytic subunit of the type 1A PI3 kinase enzyme. So there is a strong interaction here. Okay, so they're bound nicely together. Okay, now what does the NSH2 stand for? Well, SH2, let's start with what SH2 stands for. SH2 stands for, once again, SARC homology, that's the SH, okay? And then it's SARC homology 2 domain. Okay, so SH2 domain. Now, the N at the front of SH2 means that it's the SH2 domain which is closer to the amino terminus of the polypeptide. Okay, so there's going to be another SH2 domain within this regulatory subunit, and that SH2 domain is going to be closer to the C terminus of the protein. Okay, so this one is the first one closer to the amino terminus, hence it's called the N-SH2 domain. Okay, now, after the NSH2 domain, you then have this region where you have two alpha helices. Okay, so remember, an alpha helix is a spring-like structure, okay, where the polypeptide folds up into a spring-like structure, like so. Okay, so this is the first alpha helix here. Okay, and then it's going to coil back around like so. Okay, so you have these two alpha helices sort of coiled around one another like so, and this domain is the portion that the catalytic domain of the catalytic subunit of the PI3 kinase enzyme is going to interact with. It's the point that the C2 domain is going to interact with, and it's also the point that the adapter binding domain here in blue is going to interact with. Okay, now what is this portion called? Well, I'll come to its name in a moment, once I've told you what comes after this portion. Okay, so after this you have another SH2 domain. Okay, so this one is the second SH2 domain, and it's very close to the carboxylic acid tail here, so it's called the C-terminal SH2 domain, and is therefore abbreviated to the CSH2 domain. Okay, so I'll colour in the CSH2 domain in turquoise here. Okay, right. Now coming back to this region where we have these two intertwined alpha helices, like so. Okay, this domain where we have these two intertwined alpha helices, which is responsible for binding to many of the domains of the catalytic subunit here. Okay, so there's bonds here, there's bonds here, there's bonds here. This is known as the I-SH2 domain. Now, it is not a Sark homology 2 domain. Okay, what this stands for is the inter-SH2 domain, because it sits in between the two SH2 domains. It sits in between the N-terminal SH2 domain and then the C-terminal SH2 domain. Okay, and therefore it is in between them. It is inter-SH2, and therefore it's called the inter-SH2 domain. Okay, right. So that now is the complete structure then of both the catalytic subunit of the type 1a PI3 kinase enzyme and the regulatory subunit of the type 1a PI3 kinase enzyme. Okay, so this could be either a P110 alpha, a P110 beta, or a P110 delta. Okay, this, well, as I've drawn it, it would be P85 alpha or P85 beta. If you scrap those two domains there and just have the amino terminus here, then it could be P50 alpha, P55 alpha, or P55 gamma. Okay, any one of those shorter ones. Okay, and that would create as a type 1a PI3 kinase enzyme. Now, this complex is present normally within the cytoplasm of cells. Now, that's very important to understand, because this catalytic domain of the catalytic subunit is actually active. It could catalyze the reaction already. Okay, it does not need to bind to a receptor tyrosine kinase to actually catalyze the reaction. Okay, it's perfectly capable of adding a phosphate group onto phosphatidylanocetyl 4,5-bisphosphate molecules. What is stopping it from doing so is it's in the wrong location. It's within the cytoplasm where there is no phosphatidylanocetyl 4,5-bisphosphate. Okay, so a big part of activating this enzyme is merely getting it to the underside of the cell membrane so that it can come into contact with its substrate, the PIP2 molecules, and then actually convert them into PIP3 molecules. It is true that when it interacts with the receptor tyrosine kinase, that the regulatory subunit undergoes a few conformational changes, and it does activate the catalytic subunit a tiny bit, but the main way that receptor tyrosine kinases actually activate the conversion of PIP2 molecules into PIP3 molecules in the inner leaflet of the phospholipid bilayer is merely by acting as a site onto which this complex can dock. It's then localized underneath the phospholipid bilayer of the cell membrane, perfect location for actually converting PIP2 molecules into PIP3 molecules, and then PIP2 does convert into PIP3, and then it will have downstream signaling properties. Okay, right, so we'll have a break there, and then in the next video we'll discuss receptor tyrosine kinase activation of these type 1a PI3 kinase enzymes.